Hi everyone, our topic is about vaccine development, and the specific paper that we will be discussing today is on human adenovirus type 5 as a vector for vaccination against COVID-19. So one technique used for viruses in vaccine development are engineered vectors from the virus to initiate an immune response. And there are some advantages to this. One being that in comparison to using a subunit vaccine where only parts of a pathogen are used, they actually stimulate cellular responses that are far better and way more efficient in eliminating infected cells. They also promote high immunogenicity without using an adjuvant and provide long-lasting immunity, even sometimes in some cases with just one dose. They also are able to deliver virus antigens to specific tissues or cells. And it is found that in the presence of an antigen on the surface of vectors or even within the virion itself, it can also play a role in initiating an immune response. Now, there are two types of viruses that can be used for these kind of vaccines. One being a replication deficient virus, and it's just what it sounds like. It's a virus that cannot replicate. Now, these have the vector on their surface. However, their expression is controlled by an exogenous promoter. The second is a replication competent virus. So these can replicate, and they are used to mimic the natural infection that can occur in a host. So cytokines and co-stimulatory molecules will create a strong adjuvant effect. An example I wanted to talk about in regards to vector viruses is VSV and Ebola vaccines, also known as RVSV-ZBOV. It's going to be right here on the right. So the G protein encoded by the viral genome is used for attachment of the viral particle into the host cell and is replaced with the transgene of interest. You can see that on the figure right over here. The G protein coding region is right here, and then it was replaced with the transgene when using it as a vector for viruses. But in the vaccine for Ebola, this is actually going to be the Ebola glycoprotein. Now, some characteristics or reasons why VSV serves as a promising candidate for this vaccine is that it is able to replicate at high titers, it's low seroprevalence, and it has a minimal pre-existing immunity in humans.
Some more examples of viruses in vaccine development include conventional vaccines. These vaccines contain antigens derived from attenuated viruses that can still induce an immune response. Conventional vaccines take advantage of the virus's ability to stimulate the immune response even when inactivated or weakened. And a couple examples of this kind of vaccine are the MMR combined vaccine, the rotavirus vaccine, and a smallpox vaccine. Another example of a vaccine are subunit vaccines. These are comprised of subunits from the virus particle used for stimulation of the immune response. And then the efficacy of these vaccines is derived from the retained immunogenicity of viral subunits, a key characteristic of viruses in vaccine development. Two examples of this kind of vaccine include the HPV vaccine, which utilizes the hepatitis B surface antigen to induce an immune response, and also an HPV vaccine, which is actually a different type of subunit vaccine called a VLP, which is virus-like particles. These particles are designed to mimic morphology of the virus and contains the capsid protein L1.
So to give a general introduction to the paper that we'll be discussing, we'll be talking about adenovirus as a vector for vaccination against COVID-19, so the former of the methods that were described above. According to the World Health Organization, COVID has a very high mortality and morbidity rate with 7 million global deaths and 780 million global cases since the pandemic began. This figure indicates that result tracking over time. So as you can see, death rates have overall decreased over time. However, these numbers have not decreased to zero. Regarding current updates, there are around 100,000 cases and 1,300 deaths in the last 28 days, which indicates that COVID is still an issue that needs to be addressed. Additionally, this is likely due to the pandemic nature of the COVID-19 virus, which has led to viral evolution. With the infection of many hosts across many countries, as indicated in this phylogenetic tree, this leads to lots of opportunities for viruses to change and mutate, even though the Coronaviridae family is known to mutate slowly due to its error-correcting polymerase. This results in the introduction of many variants. Another reason why this is a problem, with the newest variant introduced as late as January 2025. High rates of mutations can lead to decreased vaccine efficacy and higher mortality and morbidity rates, which necessitate new vaccine formulations. Vaccine development for COVID has utilized many different methods, commonly RNA vaccines and recombinant protein subunit vaccines, but investigators wanted to see if they could use viral vectors, as these have been comparatively under-investigated. So they aim to design a adenovirus type 5 vector vaccine for COVID-19 treatment with sub-questions, should that vaccine be successful, in the efficacy of different administration routes and the type of immune response that was activated.
There are three different kinds of viruses that are used in this study, the first and most prominent being SARS-CoV-2 B1351, which is a variant containing a mutation in the receptor binding domain. This variant is also used in this study because it is infectious in both humans and mice, which is very advantageous when one is employing vaccine trials in a murine model in the hopes of eventually pursuing human clinical trials. The second virus used, our vector, lovely human adenovirus 5 or AD5. This is a great vector in vaccine development because human AD5 is highly immunogenic. It solicits a humoral response in humans, and it has a genome that is really easy to manipulate, so it's easy to work with. And the third is vesicular stomatitis virus. Now, it's a little bit out of the blue to use something like this in something that pertains to SARS-CoV-2 and 85. However, it is used as an assay. Now, to my right is a busy figure, but I'll break it down for you. This figure kind of resembles the vesicular stomatitis virus pseudovirus neutralization assay used in this paper. Now, a pseudovirus is basically a virus that is modified to contain components of another virus. In this case, the S protein from SARS-CoV-2 B1351. You can see these S proteins being made via cellular machinery and this pseudovirus budding with the VSV like body with the S protein. Another notable thing that this pseudovirus contains is the fluke reporter gene for luminescence. And that is really important because when the virus enters the cell, enters, and expresses its genome, it also makes luciferase and creates a luminescence reaction within the cell. Now, this is important because this can only happen if the virus is allowed entry into the cell. The neutralization component of this assay comes from antibodies. These antibodies all have an epitope for the S protein on the surface of VSV. And what happens in this assay is that both VSV and these antibodies are co-infecting a cell culture of cells that are permissive to the pseudovirus. And when VSV is neutralized by these antibodies, it cannot enter the cell and cannot cause luminescence. So luminescence equals no neutralization or negligible amounts, less, and no luminescence means that neutralization was employed and was successful, which is really important. Viruses in this study were also used as a vector. To my right is a figure that in a rudimentary fashion displays the AdEasy system, which was used by the people in this publication. In this system, there is a shuttle plasmid that contains the genome portion that encodes for the S protein, flanked with homologous regions, and these regions are homologous to the 85 backbone, which is an adenoviral genome derivative that is attenuated and is not fully infectious. When both of these plasmids are in a host cell, in this case E. coli, we hope that homologous recombination happens, yielding a recombinant product as seen to the right. This recombinant product is then transfected into cells that are permissive of this quote unquote infection, And in this case, we use human embryonic kidney cells, HEK293. Um, the cell recruits machinery to the plasmid, expresses it, and eventually, after assembly, viral progeny, these adenoviral vectors containing the S protein are made. They are ultimately purified and then used in further vaccine trials.
So these are the first set of results where we'll be discussing the antibody, aka the humoral response, as it was quantified using an ELISA assay for the created vaccine. There are three different administration methods tested: the intramuscular, intranasal, and intramuscular primary, followed by intranasal booster and control. So let's get into the graphs, but first let's correct their spelling of serum. So the difference between these two graphs is that the serum focuses on the systemic immune response, while BAL, bronchoalveolar fluid, tests focus more on lung immune activity localized to the respiratory. So from the serum tests, we're able to determine that levels are relatively constant for all three strains across all three treatment options, where even though the intranasal treatment has the lowest efficacy, all three treatments have a marked increase in IgG systemic titers relative to the control, indicating success. This success is reconfirmed in the second graph, but also it indicates that the intramuscular primary followed by intranasal booster produces the highest level of localized IgG. This third figure again uses BAL to study mucosal immunity, specifically with IgA, as these are more indicative of this form of immunity. This figure shows again that while not entirely significant, on average, the IMU-INU treatment administration method had the highest IgA titer production, meaning that this method of injection induced the greatest mucosal immunity. This last figure indicates the results of the pseudovirus assay that was explained prior, checking whether the pseudovirus expressing the spike protein mutated had a marked increase in neutralizing antibody titer relative to the control. From this, we see that the IMU-INU treatment exhibited the greatest relative increase in neutralizing antibody titer relative to other samples, which overall indicated that the IMU-INU treatment induced the greatest humoral response, agreed upon with all previous figures.
In order to determine the bias towards a Th1 or Th2 immune response, the researchers conducted a cytokine assay and calculated the concentrations of cytokines in the three different treatment groups. They analyzed isolated splenic lymphocytes from the three groups of vaccinated mice, and the results indicated a strong bias towards the Th1 immune response or cellular immunity. The figure on the right shows production of different cytokines when the lymphocytes were exposed to different strains of SARS-CoV-2. In A, the lymphocytes were exposed to Wuhan strain of SARS-CoV-2, and the intramuscular primed with intranasal booster treatment group exhibited the highest concentration of the Th1 cytokines, interferon gamma, TNF alpha, and IL-2. When the lymphocytes were exposed to one of the beta strains, a similar trend was seen. The level of Th1 cytokines in the intramuscular primed with intranasal booster group were higher than the other two treatment groups. This trend continued when the lymphocytes were exposed to another beta strain, where the intramuscular primed with the intranasal booster treatment group exhibited the highest concentration of Th1 cytokines. These results depicted a favorable response for viral clearance based on the production levels of Th1 cytokines. And in general, the results indicated that the group that received the vaccine intramuscularly with the intranasal booster exhibited the highest level of Th1 cytokines and produced the most robust Th1 response.
So, in summary, the vaccine was successful in inducing an immune response, and it was found that the best administration method was through an intramuscular primary injection, then followed by an intranasal booster, and that the vaccine successfully induced cell-mediated immune response that revealed increased levels of cytokines produced by Th1 cells, and therefore a Th1 skewed immune response. And here are our sources.